When I started my undergraduate studies at Montana State University in Bozeman, I was lucky to have a work-study job at the Museum of the Rockies. This was the home of Jack Horner, discoverer of the first North American baby dinosaurs, science advisor for the Jurassic Park movie franchise, and suddenly, my new boss, an academic advisor. Jack liked to test the sincerity of his potential students' dedication to paleontology, and I was no exception. My first day in the museum, Jack introduced me to my assignment. He handed me a small dental pick and a small paintbrush, and then he showed me the bone that I would be preparing using these simple tools. It was the massive thigh bone of an Apatosaurus. At over 5 feet long, it weighed so much that it couldn't be lifted up to a table to prepare. Instead, it lay in all its glory in a giant plaster jacket on a rolling dolly. My task? Remove the rock and dirt from the bone so that all the anatomical details could come to light. I spent the next 9 months of my life, in every free hour that my schoolwork allowed, and sometimes even at night, sprawled out or crouched on the floor of the prep lab alongside that gigantic bone. The work of the other preparators and students went on all around me. Scrape, scratch, brush, scrape, scratch, brush. Occasionally harden with a little glue, and then back to scraping, scratching, and brushing. Over those 9 months, I had lots of time to ponder the sheer scale of sauropod dinosaurs. And yes, many hours to curse the huge size of the adult Apatosaurus that had relegated me to the floor of the lab for so long. As I picked the sediment away from that giant bone, a question began to form in my mind. How in the world did sauropods get so damn big? By the end of May, I finally finished preparing my femur. I'd done it. Jack and the seasoned preparators in the lab had bet on me to get bored and give up, but I stuck to it till the bitter end. I proudly walked into Jack's office, excited to share my news. "Jack, I'm finished!" I exclaimed. He's always been a man of few words. His response? Nope. Now you have to flip it over and clean the other side. My buoyant mood deflated in an instant, just like air was let out of a balloon. Now, don't get me wrong, it certainly wasn't because I was over paleontology. Quite the contrary. My time with the beast had taught me something important. I wasn't destined to be a fossil preparator. I just wasn't patient enough. I wanted to do research, and I knew just what I wanted to study. "No way," I said to Jack. "I am ready for research." He glanced my way from behind a stack of books as high as the desk. "Oh, yeah? Well, if you want to do research, you must have a question." "Kristi, what question do you want to answer?" Without a pause, I blurted out the question that had been rolling around in my mind over the last 9 months. "Jack, I want to know, how did sauropods get so big?" He laughed, and with a sparkle in his eye, he said, "Now that is a great question. Go figure it out." And there begins the story of my life's work, teasing apart the different paths that dinosaurs took to grow up big or small. In this lesson, I'll take you deep inside dinosaur bones and share the tools that we use to investigate the growth rates of dinosaurs. Let's start with my beloved sauropod giants and touch on a few other dinosaurs as we fully flesh out the story of growing dinosaurs. Dinosaurs are a remarkable group of animals for many reasons, but one of the coolest is the huge range of body sizes and shapes that their adult forms take. Just consider the size differential between the tiniest known adult dinosaur, about the size of a chicken, and the biggest extinct dinosaur, a sauropod like Patagotitan. The comparison gets even crazier when we consider the tiniest living dinosaur, the bee hummingbird. Even more interesting is that all dinosaurs started out small, hatching from eggs that at their largest were no bigger than soccer balls. About like that. That includes the giant sauropods, the largest of which ends up stretching more than 100 feet from nose to tail and weighing in at around 100 tons. That's about 15 African elephants, just in case you were wondering. When the first sauropod bone, a single tail vertebra, was found back in the mid-1800s, the famous comparative anatomist Sir Richard Owen named it Cetiosaurus, or whale lizard. In just a few years' time, Owen would coin the term Dinosauria for 3 other giant reptiles. but he famously left Cetiosaurus out, assuming that at such a large body size, this creature could never have walked upright on land like the other 3 dinosaurs. Instead, he imagined that tailbone belonged to a giant marine crocodile who would need its massive body to be supported by water. Even though Owen's interpretation was wrong and Cetiosaurus was really a dinosaur, he was prescient in his choice of name. We'll circle back to this idea later on in this lesson. Unfortunately for sauropods, the overgrown lizard image would continue to haunt them for more than a century. Some paleontologists took sauropods out of the water, but positioned them with limbs sprawled out to the side like those of a lizard or a crocodile, despite the fact that the anatomy of sauropods pointed to an elephant-like body with pillar-shaped limbs. They thought this posture would have only been possible if sauropods spent their entire lives walking with their huge rib cages and internal organs hanging down into trenches, which seemed implausible. Other paleontologists kept sauropods up to their armpits or necks in water, even suggesting in some cases that their nostrils would have been perfect snorkels for underwater walking, and that the strange teeth of sauropods might have been best suited for daintily nibbling on clams. Sauropods were really the last dinosaurs to have the reputation as huge, dim-witted reptiles whose impractical body sizes doomed them to extinction. As a sauropod scientist, it is precisely their impractical, alien-like bodies that drive my interest, especially once we begin to fill in the story of their lives. All sauropods hatch from eggs that are no bigger than large grapefruits, and somehow, nearly all of them grow to enormous proportions as adults. They hit upon their weird body plan very early in their evolutionary history and stuck to it, with only relatively small variations in neck length, skull and tooth shape, tail anatomy, and body size, giving rise to the more than 200 species we've discovered. Far from being evolutionary dead ends, sauropods were among the most successful of all dinosaur groups. Though the 1980s and 90s saw the height of the dinosaur renaissance, sauropods were still kind of stuck in the dark ages. Even while museums were reorganizing their exhibits and changing poses to make dinosaurs look more realistic, paleontologists were still publishing papers suggesting that sauropods, growing at similar rates to living cold-blooded lizards and crocodiles, would have taken more than 100 years to reach sexual maturity, and even longer to reach their giant adult body sizes. Now think about it. Would it make any good biological sense to grow really, really slowly for a century before you could reproduce? No! Chances are you'd be eaten by the ferocious theropods long before you ever got a chance to mate. But if this hypothesis doesn't make great logical sense, then how did sauropods do it? How did they grow from tiny babies to huge adults within a fairly short amount of time? This is the research question that I've been asking for my entire career on sauropods as well as with other dinosaurs. So how do we investigate the growth rate in fossil animals? It turns out that the clues are preserved deep inside their bones. When we want to figure out how fast a living organism grows, it's pretty easy to do. We simply watch and measure, perhaps by documenting a change in height over time, or a change in mass over time. This is what the pediatrician is doing whenever they weigh and measure kids, charting their growth on a predictive chart of median values for gender and age. For dinosaurs, it's a little more challenging. To study dinosaur growth, we need to have skeletons that span lots of growth stages, from tiny babies to the biggest adults we can find, and all sizes of juveniles and subadults in between. This is what we call an ontogenetic series, and it is the starting point for figuring out how fast a sauropod, or any kind of extinct dinosaur, may have grown. Once we have an ontogenetic series, it is time to focus on the bones. It turns out that we're lucky because vertebrate bones serve as an archive for all kinds of data about their lives. On the outside, bones show bumps and grooves that indicate the presence of tendons and muscle insertions, which provide the key data we need to answer questions about movement. Their shapes reveal the anatomical details that allow us to determine how different animals are related, especially in the fossil record where soft parts are usually missing. Bone shapes can even give us hints about how an animal's skeleton functions. The limbs of diggers look different than those of runners, swimmers, and flyers. And the insides of bones tell us even more. Whether in living animals or fossil species, bones preserve microscopic details within that provide clues to their growth history, age, gender, body temperature, and even specific intervals of stress, disease, or injury they may have experienced during life. We'll revisit some of these kinds of details in later lessons, but for now, let's focus on the clues related to growth rates and age. The first important point to note is that when an animal's overall body is growing quickly or slowly, its bones do the same thing. When we crack open a bone and take a look at it under a microscope, we will see complex and beautiful patterns that can be used as proxies for overall body growth patterns. The study of the microscopic structure of bones is called bone histology. Histology usually means the study of tissues, and that's also true of bones. Bones are growing, changing, dynamic tissues, just like your stomach, liver, or heart. The way in which they're just a little different is because in addition to cells and blood vessels, bones also include a microscopic mineral component. Long bones start out in embryos as little cartilage models. Once they are vascularized by an artery that supplies nutrients, a group of these calcified cartilage cells die, and they are replaced by new cells called osteoblasts. Osteoblasts begin forming bone matrix, which consists of an interweaving pattern of the elastic protein collagen, which makes bones a little flexible. As bones are forming, osteoblasts also concentrate the mineralized crystalline component that gives bones their rigidity and hardness. These crystals are made up of calcium phosphate hydroxide, also known as hydroxyapatite. Collagen molecules and hydroxyapatite crystals line up in parallel as bones grow. As bone growth continues, the osteoblasts become trapped, along with blood vessels that are helping to supply that developing bone with nutrients and remove waste. Once trapped, these osteoblasts change their role to become bone-maintaining cells rather than bone-building cells. They're now called osteocytes. This type of bone is called primary bone, and its organization documents the relative speed of bone deposition. There are several signs that we look for to assess how an animal is growing. The faster an organism is growing, the more disorganized its primary bone matrix will be. The more abundant the osteocytes, the faster a bone is being deposited. The higher the blood supply, the faster a bone is growing. This means that more blood vessels are traveling in more diverse directions within a bone, some going around in circles along the circumference of the bone, some radiating out from the center of the bone like the spokes of a wheel, and some traveling up and down the long axis of the bone and intersecting with each other in an interweaving network. In slower growing bones, mineral and protein have time to align themselves in tiny, thin, parallel layers. There are fewer osteocytes and less blood supply. Now, you may already know that the soft, squishy, microscopic protein molecules, cells, and blood vessels aren't preserved in dinosaur bones. Lucky for paleontologists, the accompanying spaces within the crystalline matrix where these soft parts sat when the animals were alive are preserved. This means that we can directly compare the primary bone structures of living and fossil animals and extract similar data from each. Sometimes in slow-growing animals, we observe highly mineralized rings within the bones that point to complete cessation of bone growth for a period of time. These rings are called lines of arrested growth, or lags. They are most common within the bones of slow-growing, cold-blooded animals who must pause their growth seasonally during the cold times of the year. Lags also occur in fast-growing organisms, sometimes because of some external stress, like drought, and then once the animal reaches adult size. We have put all these different things about bone growth to the test in living vertebrates, including birds, mammals, and reptiles. Specifically, we've investigated whether bone growth rates accurately reflect overall body growth rates. and if particular microscopic patterns in bones could be linked to a specific growth rate in living animals. It turns out that we can do both. So we're able to pinpoint a range of possible rates for different patterns of primary bone growth. Generally speaking, among living animals, reptiles grow slowly, mammals and some birds have growth rates that overlap, and other kinds of birds grow faster than any other living creature. This means that we can measure an amount of primary bone in a dinosaur fossil, compare its microscopic organization to living animals, and determine precisely how long that dinosaur bone took to form. So, let's pause here and do a little test of your newfound bony knowledge by looking at these signatures of growth in some thin sections of fossil bones. First up, in this image, you can see lots of little squiggles all over the place with different orientations. These are the spaces where a rich network of blood vessels supplied this fast-growing bone with nutrients. Look even closer and you can see tiny little black specks. These are the spaces where bone cells would have been when the animal was alive. If we could zoom in even closer, we would see that the preserved microscopic minerals are also crazily organized. That's indicated by the pinks, blues, and yellows that you can see in this image. Taken together, all of these features point to a relatively speedy growth rate. On the other hand, this image has hardly any of those blood vessel squiggles, very sparse, tiny spaces for bone cells, and lots of lines of arrested growth. Even at the tiniest microscopic level, the minerals that made up this bone are super organized, creating these lines that you can easily see. All these features indicate that this bone was growing much more slowly than the previous one. Right now you might be saying, that's all great, but what in the heck are those lifesaver-shaped things? What do they have to do with growth? Those are great questions. Now that you're an expert at primary bone tissue and what its general features mean about the relative speed of an animal's growth, we need to introduce one more bone type, secondary bone. Faster-growing animals need to continually remodel their primary bone tissue, so they use their own bone mineral to do so. Sometimes they'll need to repair a microfracture, or they'll need to grow some new bone to rapidly accommodate changing body mass or behaviors. Other times, they'll need to use their own bone mineral when shelling eggs and when growing skeletons for their own offspring. Here's how it works. Cells called osteoclasts travel around the blood supply and begin to dissolve the bone around the blood vessel they're traveling in. This causes all those minerals and protein components to enter the bloodstream so they can be moved to places where they are more needed in the body. Nearby osteoblasts move into the newly opened spaces left behind and begin to redeposit new bone. This process results in a characteristic lifesaver shape that we call secondary bone. In older animals, many intervals of remodeling have occurred, erasing the original bone and leaving behind many overlapping generations of secondary bone lifesavers. If we take one more look at our two bone examples, you can easily see that the faster growing bone also has more of the lifesavers that signal remodeling. Remodeling is exactly why we need to look at the ontogenetic series of dinosaurs when we're trying to understand how fast or slow they grew throughout life. Usually, big adult dinosaur bones have almost all their primary bone tissue completely remodeled by secondary bone. If that's the case, all we can say is that the bone has been remodeled. As we sample smaller and smaller individuals of a single species, we're essentially looking back in time to reconstruct what the primary bone looked like before it was remodeled in younger and younger members of the group. Cool, huh? When we study dinosaur bone histology, it's kind of dramatic. The first step is identifying your ontogenetic series. Ideally, we will study several different bones throughout the series that represent different functional environments, since we know that bone microstructure is driven by many things. In my own study of the sauropod dinosaur Apatosaurus, I sampled several different arm and shoulder bones for each of my ontogenetic stages. In the forearm, the radius is a non-weight-bearing element, while the ulna is the weight-bearing element. The scapula, or shoulder blade, forms in a slightly different way than those two forearm bones and gives us another point of comparison. We expect that each of these bones will show some differences, but we're looking to see if there are consistent patterns within each that will allow us to make an assessment of growth. Then the drama begins. We must cut the bones open in order to look inside. We usually use the mid-shafts of long bones to take our samples because they preserve the longest records of growth. Sometimes we'll take only a wedge out of the bone using small hand saws with blades lined with diamond dust. At other times, we purposefully and carefully break the bone at an existing mid-shaft crack so that we can take a chunk out and have rough edges to glue back together. This allows for the preservation of total bone length. Occasionally, we get lucky and a curator will allow us to cut the bone completely in half, usually using a high-speed diamond edge saw akin to a tile saw. That's always a little nerve-wracking. We then embed this chunk of bone in plastic, vacuum out all the bubbles, and make very thin sections using a slow-speed, diamond-edged, water-cooled saw. The wafers get glued onto glass slides and are then ground down by hand on a lapidary wheel lined with diamond grit paper. This is tough work. As you grind away the layers of bone in plastic, you're also grinding away your fingerprints. After years of doing bone histological research, I think I'd be a great criminal. I'd be untraceable. No fingerprints. Our goal is to grind down carefully and layer by tiny layer until light will pass through the bone when we place it underneath a transmitted light microscope. That's when the magic really begins. Bones under a microscope are simply beautiful. In the opening of this lesson, I told you the story of when I began my work scraping, scratching, and brushing that Apatosaurus bone. At the time, paleontologists assumed that sauropods were like overgrown reptiles, growing slowly and steadily for more than 100 years, and maybe growing forever at this slow pace until they died. I sampled a whole bunch of bones, spanning juvenile to adult Apatosaurus onto genetic stages. Unlike the bones of reptiles, these bones were highly disorganized, densely vascularized, and without any lines of arrested growth until they were humongous. Throughout their growth, remodeling was pervasive. I was able to use our comparative quantitative data from living animals to determine that Apatosaurus would have grown from juvenile to adult size. That's about 75 feet long, about 60 to 70 tons, in about 20 years. That is no reptilian growth rate. These shocking results turned the old ideas of reptile-like sauropods completely on its head. To dig deeper, I teamed up with a colleague named Greg Erickson. Together, we compiled data from the published literature on a whole bunch of other dinosaurs to see if we could figure out if dinosaurs had their own unique standard growth rate, like others had observed for fish, amphibians, reptiles, and different bird or mammal groups. Or did some dinosaurs grow like reptiles, some grow like birds, and some grow like mammals? We generated growth curves for dinosaurs small and large. We included dinosaurs that were closely related to birds and ancient dinosaurs at the base of the family tree. We included herbivores and carnivores. Our work resulted in the identification of the maximum growth rate that these diverse dinosaur species could achieve. Our growth curves allowed us to determine how long dinosaur growth spurts might have been and how many pounds they were packing on during these intervals. What we found was that all dinosaurs grew faster than reptiles. As a whole group, dinosaurs grow most like slower-growing birds and mammals. Even cooler, not every dinosaur grows the same way. Tiny theropods closely related to birds actually grow the slowest among dinosaurs, only 2 times faster than reptiles, which means that birds innovated their speedy growth rates independently. Large sauropods like Apatosaurus grew about 50 times faster than living reptiles and put on about 50 pounds per day when they were growing at their peak. This may seem outrageous at first, but these numbers are right in alignment with the other biggest vertebrates of all time, the whales. Even though Sir Richard Owen perpetuated the idea of sauropods as overgrown lizards, he wasn't really that far off. Remember, he named the first sauropod ever discovered Cetiosaurus, or whale lizard. They may have been scaly on the outside, but underneath those scales, they were more like whales in their speedy growth rates and maximum body sizes. I hope that you've enjoyed this little foray deep inside dinosaur bones. Bone histology has become one of the most powerful tools that paleontologists have at their disposal for investigating questions of growth and function. Pretty much every single dinosaur group has now been studied histologically, and the patterns that we've talked about here hold up. Dinosaurs were fast-growing animals that frequently and pervasively remodeled their bones. They stopped growing when they reached adult size, which was also generally coordinated with sexual maturity. And they grew more like mammals and birds than like any reptile. We'll touch on histology a few more times in our course, as it plays an important role in rediscovering the age of dinosaurs.